Okay, so we'll start off by defining homeostasis, which is a state of steady internal physical and chemical homeostasis is a state of steady internal, physical, and chemical conditions that are maintained by living systems/organisms. Okay, so we're going to talk about something called the reflex control pathway. So I like to say that has 5 different steps. So the first one is that a receptor will receive a stimulus. This receptor will then send electrical or chemical signal to the control center. Control center knows what the set point is. And the set point is how we maintain homeostasis. The set point refers to the set point is a range in which the organism can still survive and maintain homeostasis. Nose set point. Then we have what's called the efferent pathway, which is a signal from control center and the effector which will bring back to set point and I'll do that depending on where we're at in comparison to set point. For below set point or above set point, the effector will change it as would be needed. So next what we're going to be talking about are what's called negative and positive feedback loops. That's falling down. Okay. So I'm going to use green for negative feedback loop. Feed. Let's rewrite that. Feedback loop. So negative feedback will bring us back to set point. Positive feedback sorry, that was the wrong color. We don't want orange. positive feedback will take us away from set point. We also have another pathway called inadequate negative. It's a subcategory of positive feedback. This is where effectors are working, but it's not enough to bring back to set point like a negative should. Okay, so I'm gonna erase this and keep it our same colors. Inadequate negative is gonna be orange. Positive is red, and then negative will be green. So an example that I'm gonna use is body temperature. So, the average temperature of a human, we'll call this time, we'll call this degrees Celsius. So typically, there's a range of temperatures in which a person will go throughout a day. The average of those is usually 96.8 or 37 degrees Celsius. Sorry, that's supposed to be a straight line. Positive feedback would be anything in this red that would take us away from that. The green, you can see with these humps in this line, it's us trying to maintain set point. There'll be small deviations, but nothing extreme like here. Now, an example of positive feedback, or rather an example that we could use would be with negative feedback, is if we decided to go out in the middle of winter without any clothes on. It'd be kind of silly, why not kind of be very silly, and immediately what our body would do to be able to try to get back to set point from that stimulus is we would burn calories. This would increase our body temperature due to the breaking of release of heat and breaking those bonds between those molecules. Another option, what they would do, or what you could do to be able to be an example of a negative feedback loop, would the body would begin to shiver. Shivering will cause the person to be able to give off more body or produce more body heat and be able to take you away from being cold. It'd warm you up essentially. But all these wouldn't be as effective as using our good old noggin and going back inside to warm up. But yeah, so that right there covers our positive and negative feedbacks. Another example that we can use is blood pressure. I'm going to draw this in the center of the graph, or not graph, this diagram. Blood pressure. three main things that we attribute to blood pressure. One is peripheral. I definitely spelled that wrong. Resistance. Another one is heart rate. Another one is blood volume. These two, it's a direct effect between the two. So our heart rate increases, so does our blood pressure. As our blood volume increases, so does our blood pressure. With peripheral resistance, it's a little bit interesting. If we increase peripheral resistance, I'll label or disregard that of PR. We will increase vasoconstriction, which will in turn increase our blood pressure. The opposite, if we were to decrease peripheral resistance, then we would increase vasodilation, decrease vasoconstriction, and also be able to increase blood pressure. As you can see, positive in essence, what medicine does is it corrects things that take us away from set point. Most medicines operate under a negative feedback loop. And a good example I can think of with a positive feedback loop would be childbirth. Childbirth increases oxytocin, which will increase contractions, which will then in turn increase oxytocin more. And then it follows that cycle. Deviations from set point with positive feedback. If medicine's not able to keep this positive feedback into check, an individual can die. It's very important to understand how to mitigate or mediate between positive and negative feedback, and to be able to ensure that individuals are healthy. So moving on to the second part, we're going to be talking about diffusion. Oh. Good. So diffusion, I don't like that marker. Diffusion is movement of ions from high to low concentrations inside and outside of the cell. Okay, so we're going to erase this definition here. We're going to paint out a diagram to be able to explain all the different ways that ions can move into and out of cells. So, first thing to differentiate is this first question. Does movement require more energy than just kinetic? Start off on this side. So if it doesn't require more energy than just kinetic, we'll break it down into two other parts. And the second question we'll ask, does it require help? We'll find out a little bit more. If we're going no, doesn't require help, then we would just simply call this simple diffusion. Simple diffusion is gradient dependent. And moves non-polar small particles. Examples of this include oxygen and carbon dioxide. If an ion requires help to be able to go into the cell but does not require more energy than just kinetic, we call that facilitated diffusion. Which there are, actually, before we go into there, with simple diffusion, its rate is a linear relationship between its gradient and its ability to be able to pass through the cell. Examples of facilitated diffusion first are aquaporins. Aquaporins are the way in which the cell is able to have water going through it. It's a transmembrane protein channel, and it's specific. What you'll notice with a lot of these channels is that their specificity is high, and each channel is specific to an individual ion. There aren't channels that can work for two different channels. Another example of facilitated diffusion would be voltage-gated channels. Some top players that come to mind are the voltage sodium and potassium gated channels. They're important in a lot of different cells, but it's definitely neurons as well. Another is ligand gated. So what we can see here would be we have the sodium ligand channel. Ligand meaning that the protein, or I guess the channel, it's most of the time closed like this, if this is the membrane. Or part of the membrane. So it's usually closed, and I'll have this position right here that we can call the active site. A ligand will bind to it, and that will cause a conformational change in this protein to to open it. So then whatever substrate we want, or whatever ion we want to go into it, can be able to go in. Another example. So we have aquaporins, voltage gated, ligand gated, and the one is mechanical gated. We also have leak channels. Which can or cannot have, which can also be open or closed. On the topic of specificity, question can arise where we have sodium and potassium. Now, channels are specific to both size and charge. This is due to the proteins, or I guess rather the proteins, the amino acids that are lining down the channel or down through the channel. This isn't a really good picture, but this would go all the way through. So there's different amino acids that are all the way around here. And those interactions will allow very specific ions to only pass through. So that raises the question, we know that potassium is larger than sodium, so then makes you think, then why can sodium not also pass through a potassium channel? And a big reason for that is because the amino acids that are with a potassium leak channel or any potassium specific channel, they will remove the hydrogen sphere or hydration sphere around the potassium to allow this potassium enter, but the sodium to not, which is pretty cool. The rate of facilitated diffusion channels, it is hyperbolic. We can see that saturation does occur. Saturation refers to, or it's a limit that can be, that is approached. I'll draw it right here, that little dotted line would represent the limit that occurs when all these channels would be past their capacity because they already have as many ions passing through as possible. So unlike with simple diffusion, the greater the gradient, then the more particles they'll pass through. In this one, there is a cap which comes due to the saturation of these channels. So once the channels become saturated, then it doesn't matter how strong the gradient is, it'll remain the same. Another flavor of, if we're going to answer this question is yes, that does require more energy than just kinetic. The first example that we have would be primary active transport. Primary active transport, it requires ATP, adenosine triphosphate. A good example of primary active transport that we've talked about in class is the sodium, potassium, ATPase pump. So this pump has, how it works is 3 sodium are pumped out of the cell, while 2 potassium are pumped in. Wait, scratch that, that's not right. So, it's actually 3 sodium pumped in from the extracellular fluid, and 2 potassium are pumped out in the intracellular fluid. Now we see this happen is the sodium potassium ATPase pump has 2 conformations. In the E1 state, you'll see that an organic phosphate will bind to the molecule or to the pump. We'll draw it like this, a little lever door. So this is the ECF. This is the ICF. So when this phosphate is attached, we'll have 3 sodium that bind. And when that phosphate's removed, it'll go into what's called the E2 position, which this will cause a conformational shift where the sodium will be dumped inside the cell. Then this will free up, allowing for potassium to bind. And when that phosphate's released, then the potassium is taken out. Phosphate releases, and the potassium falls out into the extracellular fluid. Second form of channels that require more energy than just kinetic would be the secondary active transport. Secondary active transport will hijack energy off of the reactions from primary active transport. Secondary active transport, we start seeing these things, or I guess the movement of the ions in two different ways. The first one we call symport, which is where 2 ions will move in the same direction through a channel. And antiport, which is where, if we're going through a channel in a cell, 1 ion will be exiting the cell, while the other ion will be entering the cell. The example we can see with this is some instances of a sodium channel where calcium, that's a 2+, will be able to use that same channel as an antiport secondary active transport channel. Another instance that we can think about would be competition. The specificity of a lot of these channels requires very specific ions to only pass through them, but there are some instances where competition is necessary between the two. For instance, there are some channels that are they work for both sodium and potassium, but due to sodium having a higher driving force, which I'll explain in a later video, it essentially becomes a sodium channel. But yeah, that should be it for this question.
Okay, so first we're going to explain the process of osmolarity. So osmolarity is the process of solvent, which is usually water, semi-permeable membrane. That's semi-permeability via phosphoporins. Because semi-permeable membrane from concentrations. So this difference from molarity. So molarity, we're talking about moles of the solute. Here's a solution, which is not the same as osmolarity. Particles that contribute to changes in osmotic pressure. The units, molarity, like I said earlier, it's moles per liter. We usually call it big M. Osmolarity is osmoles per liter. Now, when we're talking about my sock, here's my sock. We're gonna use some stuff. So now we're going to go into osmolarity versus tonicity. So osmolarity, I'm going to draw a little diagram. So the little circle is going to represent a cell. This is going to represent a container. So osmolarity is when we're comparing two solutions. Or we're comparing amount solute in between two solutions. With the tonicity, we're talking about the effect one solution has on the other. Okay. So when we're talking typically what a cell has, okay, to be able to compare these two different solutions, we use the prefixes hyper, iso, and hypo. Hyper means more compared to other. Iso means same as other. hypo less than. Okay. So we're talking about isotonic solutions. We're usually, or isotonic, not necessarily isotonic, but isoosmotic solutions we like to use 300 milliosmoles per liter. And something that is 300 milliosmoles per liter is 0.9% saline or sodium chloride, and then also 5% dextrose. So I'll talk about these two a little bit later. So back on the topic of osmolarity, if these little dots represent solutes, these two solutions are isoosmotic. Going over here, we'd call these two isotonic. So if we were to do this, we would say that this solution is hyperosmotic compared to this solution. If we did this, we would say that the cell is hypertonic compared to its solution. So when we're talking about tonicity and the effect of those other equations, we have to take into account the permeability of these compounds. So first trial, we consider 300 osmoles for an isoosmotic solution. So we need to change up some colors. We're going to use green for our 0.9% saline. And I'm going to use red for our dextrose. And then, I don't think I mentioned this earlier, but dextrose is pretty much just glucose. Got to give it a different name when it's going through an IV. Draw our containers again. That's not what I wanted to draw. Start here. So something that's want to touch on is a big thing of how something could be. For instance, we're talking about dextrose. You look at both of these; they have the same amount of solid in both solutions, so we would call this isoosmotic. But if we did the same thing over here, what would happen is we have to remember that the cell is able to, I guess we have to talk about permeability. So, sodium chloride at resting, just at normal states, when the cell's at rest, is not permeable. It's not able to enter the cell without other help in changing different factors. Dextrose, on the other hand, is permeable. So, in this example, this changes things. So, looking at this, we would say, oh, that is isotonic. But because it is permeable to the cell that we see, I'm not going to over my thing. So our dextrose is going to go into the cell, which then makes the cell in a hypotonic solution. Water will flow in to balance osmotic pressure.
So this is a very important idea. If you were to mix up tonicity and osmolarity, you could kill someone by, or maybe not kill somebody, but you could hurt them pretty badly if you were to give them the wrong thing while you'd be trying to hydrate them and then not realize that you're creating an environment that will cause either their cells to shrivel and then cause more issues than you would have wanted, or them to what's it called? Swell. They could burst because, as we all know, malignant cells don't have cell walls. Nice important thing to think about is that water follows wherever the solute goes. Water will follow in that same direction. So that's how we can be able to tell if the cell will shrivel or if it will swell. It all depends on the interaction between the two, or the tonicity of the two solutions. So moving on to part 2 of this question, we're going to be talking about RMP, or the resting membrane potential. Okay. So resting membrane potential, if we were to draw here a cell, there's 3 big contributors that come into play when it comes to resting membrane potential. First of which is the movement of ions, separation of charges. Sorry, not movement of ions, leak channels. ATPase, sodium potassium pump comes into play. So to represent the sodium potassium pump, I'm going to draw ATP, and then we'll draw 3Na plus, 2K plus. Okay, so the sodium potassium pump is able to establish the equilibrium gradient just by changing the charges between the two, and potassium leak channels are crucial with it. Typically, on the inside of a cell, just because of how it's set up, we find that chloride is inside of the cell, and the proteins are typically on the inside of the cell. There's protein particulates that are negatively charged. So we have this separation of charges, which will affect the driving force of different ions, which we'll talk about at a later time during this video. But before I get too hard on myself, so we also have potassium leak channels. I guess potassium is going to all that. Leave. And yeah. So typically, at resting membrane potential, if we're talking about neurons, the cell will be at negative 70 millivolts, which just can be changed depending on what kind of things are. Actually, let's move on to that. So things or factors that can, factors and conditions that can change resting membrane potential are first, permeability. Second, concentration of ions. And then temperature can also play an effect.
So we'll go first with permeability. So at resting membrane potential at the negative 70 millivolts, negative 70 volts, a significant amount of sodium will be able to throw off the charge within the cell. Sodium is non-permeable, but changing the charges of on the compared to the inside and outside of the cell, this can make sodium and other ions. It can change its permeability to make it easier or slower to exit the cell. Another one is the concentration of certain ions on the inside versus outside the cell. So in the case of sodium, it doesn't really matter changing the concentration on the outside of the cell. And with potassium, because if you were to change its concentration on the inside, it doesn't really make that big of an effect because it just makes the potassium want to stay inside of the cell more due to its driving force. But if you were to change the concentration of potassium outside of the cell, that can make a greater effect to either making the cell hyperpolarize more or to depolarize more. And the difference between these two, if we were depolarizing, then we would make this negative 70 millivolts less negative, like you could say like negative 60. But if we're hyperpolarizing, we'd make it more negative, like negative 80. Another thing that can affect factors and conditions of resting membrane potential would be temperature. So changing the temperature too drastically can cause cell membranes to pretty much fall apart. Thankfully, through trial and error, I would say, well, not trial and error, but God was able to be, was very smart, and we have a compound within our cell membranes called cholesterol, which at higher temperatures, it will make the membrane more rigid. And at lower temperatures, it make the membrane less rigid or permeable. So if it wasn't for cholesterol, then be changing the permeability of these ions drastically, because we have all these different channels that are around here, and they're anchored in dependent upon how strong our membrane is. So as soon as you start introducing temperature changes, and if it becomes too drastic, that cholesterol is not able to compensate for it. What can happen at especially higher temperatures when it becomes more flowy. These protein channels and pumps and everything else can literally fall through the membrane of the cell, just falls apart and pretty much dies. So yeah, these are some different factors that can be able to change resting membrane potential. And finally, we will talk about the Nernst equation and driving forces. So, Nernst equation is equal to E ion, which is going to be equal to our negative 68 all over Z log of the concentration in, concentration out. So just breaking this up, this is the cell's equilibrium constant, or equilibrium drive. The EI. So this will tell where the ion wants to go. These related to its charge, the charge of the ion. And then this number here is just a constant. It comes from the temperature multiplied by the gas constant. And then this right here is referring to the concentration inside versus outside of the cell of that particular ion. Okay. Then we have what's called driving force. Driving force explains where the cell will want to go, or where the ion wants to go. So E ion is not necessarily this is where it's at. Okay. So driving force follows this equation, which is Vm, which is the same as the resting membrane potential minus Nernst's equation. And we can be able to see through this equation where, depending on what the conditions are, where the ions want to go, and then be able to predict how that would change things within the cell. So yeah, that's it for this one. Okay, so starting off, we're going to discuss what an action potential is. An action potential and the electrical neurons cells due to movement places. So action potential is an electric signal that occurs on neurons and muscle cells due to movement of ions in specific movements. Characteristic of an action potential is a all or nothing phenomenon. And there has everything go according as it should, or if it doesn't, then we don't see an action potential occur. It is also one directional. Signal is only transmuted down an axon, which if we're looking at just a really rough drawing of a dendrite. So this right here we would call the body. Little dots nucleus, these are dendrites right here, soma, and this is the axon. So they only go down this direction. The signal doesn't go back that way. All or nothing, one direction, or one directional. And yeah. So let's go through the different phases of an action potential. So I'm going to draw one out, and we'll go from there. Here we go. So just a representation, since it's usually with, action potential, usually think of that. Here, so when that occurs, so it's Rm, rest memory potential, assuming that balance is changed value, will be at negative 70 millivolts. So, and then we'll say threshold, negative 55 millivolts. So, first part of it is we call this area, which I'll label A, as the rising curve. And this happens due to ligand gated sodium channels opening. For that, we have the resting membrane potential established by the potassium channels. This dotted line right here, we're going to call threshold, where as soon as right here in this B position, B is call the action of the cell reaching threshold, which equals opening of voltage sodium channels. So when we first open the ligand channel, we can see that the cell will depolarize here a little bit, but then it'll super depolarize as soon as we hit threshold. And at this point, we'll label this C. So our sodium voltage will close, and our V, our voltage potassium gated channels will open, which if the cell is unmyelinated, it would do this. So this occurs because we have mass exit of potassium ions leaving the cell, which will cause it to polarize even below our starting point, which is what we see right here. This point right here, our voltage gated potassium channel, this will close, and then we'll establish to where we're supposed to be at the refractory period with the potassium channels. That's how the cell is able to get back to its starting point it needs to be. This red area, this is what's called the absolute refractory period. So in this red area, anytime through that, it's impossible for us to get a second action potential. This is because the sodium voltage channels are inactivated. So because they're inactivated, there's no way to reach threshold because you're not able to reopen them. So labeled blue is called the relative refractory period. We could get another potential action potential if the stimulus is strong enough. That has to be a strong enough signal for it to be able to occur. So yeah, these are all the channels that are involved with the different stages of the action potential. On. So we'll compare these two different aspects, which is the graded potential versus action potential. So graded potential, it could be depolarization or slight depolarizations or hyperpolarization of the cell or of membrane. These are marked by their amplitude. And if you get enough of them together, then it can lead to an action potential. Action potential, on the other hand, like I was saying earlier in the video, it is an all or nothing. And we see extreme depolarization of membrane and hyperpolarization. So the relationship between these two is with the greater potential, if you get enough of them together, then you can get an action potential. But the opposite is not true with an action potential. Action potential will not lead to graded potentials. Another concept that we can talk about is capacitance. So capacitance refers to, actually, let's start here with the length constant. So length constants, we use the this Greek letter. I believe that's gamma. I think. Yeah. Maybe that's gamma. Maybe it's not. Anyways, so it's the square root of Rm. Alright. So this is internal resistance, membrane resistance. Length constant, we can also attribute it to, I think Brother Shaw likes calling it Blanc. So this is the, Brother Hunt will call it a sphere of influence. So this is what travels down the axon. And this is important with, once X potentials reach, then this causes a chain of events to occur. So capacitance is, we call our charge over volts. So it's described as. And then our time constant is 63, is the time that it takes to reach 63% of our expectation. Time it takes to reach 63%. The action. Okay. So there's other ways that we can be able to make cells, or I guess neurons, a little effective. One way that squids will use is they increase the diameter, which will decrease Ri and increase the Rm, forming a stronger length constant. Because when it comes to action potentials, the strength and duration of strength versus duration of signal are inverse. So the stronger your signal is, then the weaker, shorter the duration, and vice versa. So a way to overcome that with that humans do and a lot of other animals is that we'll put myelin. So I'm going to draw another neuron. Neuron. So to increase its capacitance, or how flowy your membrane is, what happens is they put myelin around. So what myelin will do for the cell, so if we draw an action potential again, right here, negative 55. So the cell will do its thing as normal, but the difference is it doesn't hyperpolarize as much because this myelin makes it more difficult. Well, not more difficult, but it spreads out the charge of the potassium that's going to want it to more than likely to enter back into the cell because there's so many, say our leak channels right here. There's so many positive charges that are right here, and it's so spaced out over here that this will repel it right back into where, back into the cell. So the myelin will decrease the um, area, the area of the membrane, and change its distribution of charge. Then the potassium will enter back in the cell and that hyperpolarizes deeply because we have so much potassium leaving the cell. Myelin makes our neurons a lot more effective and be able to pass on quicker signals. So in the next part, just keeps moving. So we're going to be talking about the structure of the chemical synapse and explain how signals pass from pre to post terminal. So I'm going to zoom in on it. So here's my dendrite. This right here is my presynaptic dendrite. Draw like this. That line right there, because that makes it like it's blocked off, which is not. Yeah. So this is the presynaptic dendrite. This area in between these two dendrites is called the synapse. This is the postsynaptic dendrite. So our action potential will go down this way. Here we have our bond. And this bond will cause for these right here we have what's called a voltage calcium channel. And this sphere of influence or bond will cause for calcium with a conformational shift in this. Calcium will enter in to the cell or to this neuron, and then that will cause chain reaction of where these vesicles, which are filled with neurotransmitters, to go through a process of exocytosis and bond with this membrane here, and then they will enter into the synaptic cleft. So over on this opposite side of this neuron, or on this other neuron, that postsynaptic dendrite, we have different receptors. One that I'll use right here, just for these ones. Actually, no. So these receptors will react with these neurotransmitters will bind onto, for on the postsynaptic, they have these areas where neurotransmitter can bind. We call those the active sites. When these neurotransmitters will bind, they'll cause a signal to go down this this neuron, and depending on what the receptor is, it could cause an excitatory inhibitory response. Which this can be dependent upon, because this is like a super simple way of doing it. How it really work is there's that's wrong, hundreds of neurons that will gonna draw 6 of them. There's tons of neurons that will cause this to happen. So there's different ways to be able to figure out if something would be excitatory, inhibitory signal. It depends on what this receptor is going to transmit down the cell. Like for instance, if this neurotransmitter calls for a closure of potassium, so this is potassium voltage channel, it causes it to stay closed, then it'd be an inhibitory response. Or if it was allowing chloride to go into this neuron, it could be inhibitory. Or if it's excitatory, it's sodium entering or something similar. And something that can be able to help ensure whether or not it's an excitatory or inhibitory response would be one is its temporal and what's it called? Spatial. So we're saying it's temporal. The simple effect, we're talking about the number of, it's the frequency of signals. Well, spatial effect, it's whatever's closest to the receptor. So this receptor, this synapse right here, would have a much larger effect. And say this one over here, because of that spatial location. And also number of receptors. That's the receptors present. Or that's something. That's some of the ways that it would, how these signals can be able to pass down through there. The example we talked about class was the NMPA receptor. PA receptor. So in the example of this, we're going to say their neurotransmitters are glutamate. So glutamate would go and attach them to here. This receptor stays closed, but is a calcium receptor, which would be an example of an excitatory response. When this glutamate binds to this receptor, the AMPA receptor, it'll cause a Blanc or sphere influence to be able to travel down here. And if it's strong enough, what it can do is it can knock out this magnesium, cause a conformational drift within here. And then calcium can be able to enter into that neuron and be able to create an excitatory response. But yeah, that's all I got for this video. Okay, so we're going to compare and contrast the sympathetic division, parasympathetic division, somatic anyways, yeah, go through that. So starting off sympathetic, parasympathetic. Number of neurons, one. Sympathetic and parasympathetic both have two neurons, a pre and post. This will innervate directly to its target. Location of ganglia. For the sympathetic, it has different kinds or different ones. So it will go to sympathetic chain ganglia after it exits the neck. It'll wrap around itself, synapse of the sympathetic chain ganglia, and then go to Durgan. That is from T6 and above, or T5 and above. For a blank neck or splank, yeah, splanchnic nerve, which is going into T6 to diaphragm. So go to a synapse on a lateral chain ganglion. There's another type called adrenal medulla pathway, where this won't synapse anywhere except for directly onto the adrenal gland, which is important because this will lead to the production of epinephrine and norepinephrine, which are not epinephrine, which both of these are important for our adrenergic receptors, which we'll talk about later. For the so this autonomic will come out of the lateral horn of vertebrae, and the somatic, depending on if it is sensory, motor. If it's sensory, it will come out of the distal, and if it's motor, it'll come out of, or not distal, sorry, posterior. If it's motor, then it will come out of the inferior. Okay. Cool. Moving on. So at the sympathetic, typically has a long, sorry, shorter preganglion, and then it kind of depends wherever the target is of how long the post is going to be. So if we're drawing our typical sympathetic, sorry, that's the other way around. Long pre, short post. Long pre, and then with the post, it just ignore me. It just depends on where the receptor's going. So between this and then our terminal ganglion, we have what's called an N2 receptor. It's a cholinergic receptor. So it'll deal with acetylcholine, which we're going to label as ACH, so be acetylcholine. And then it'll go to a different receptor, and like I said earlier, its post is going to be unmyelinated. And sympathetic will always go to smooth muscle, cardiac, and glands. Now its post receptors, they're going to be different. They're atrogenic. So they work with their ligands, I guess. So these are G protein receptors. Yeah. And they go from alpha 1, alpha 2, beta 1, beta 2, beta 3. And not all receptors have, or yeah, not all of the neurons have all these receptors. On the same one, there'll be one that's predominant and, or yeah, is the dominated one or most frequent one that's present depending what the tissue is. Okay, and then cool. Yeah, let's move on. So sweat glands, they're not part of the parasympathetic. They're still sympathetic. So this one's called an N1 receptor. It's still cholinergic. So acetylcholine is going to be the neurotransmitter between the two. Still dotted line, very short. These are going to be super short to be able to get in there, and then they innervate to common sweat glands. So this is M1, which is muscarinic. These are also cholinergic receptors. Okay, then with parasympathetic, it's gonna be a little bit different. So still myelinated. Still gonna be an N2 receptor between these two. Still cholinergic. The difference here, it's gonna be same thing, smooth muscle, cardiac, glands. But we got M1, M2, M3, M4, and M5. So the difference between parasympathetic and sympathetic. Parasympathetic, the pre is going to be cholinergic receptor with an adrenergic post receptor. With parasympathetic, both the pre and the post are cholinergic receptors. Something that the norm or like the 99.9% rule to live by is if the number's odd, it's going to lead to an excitatory response. If that receptor is activated with an agonist, vice versa with an antagonist. And then even if that receptor is turned on, then it's going to lead into an inhibitory. So we're going to erase all this. I'll redraw them all out. Go from there. Make a different little chart. Okay, so we have Isoenergic going to be alpha 1, alpha 2, beta 1, beta 2, beta 3. Cholinergic is our N1, N2, M1, M2, M3, M4, and M5. So there are some differences between these two. It's what their secondary messenger ion is, which can be one of two. It's either IP3 or CAMP. So the receptors where they're secondary receptors IP3, increase in IP3 will relate to an increase in calcium being released. So N1 and N2, they're in their own group. You know, have secondary messenger ions, at least right now. These ones are M1, M3, M5, alpha 1. For CAMP, alpha 2, M2, M4. So we got 1, 2, 3, 4, 2, 3. 1, 2, 3, 4, 5, 6, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh, sorry. And 1, and 2, beta 3. So what's interesting about beta 2 is associated with lux. Typically an increase in CAMP little c, big A, and B. The rest of these, a lot of them are inhibitory, but with beta 2, we increase the CAMP, and that leads to an inhibitory response. Or, sorry, typically, when we increase the CAMP, then that'll lead to an excitatory response. But with beta 2, the lungs, an increase in CAMP leads into an inhibitory response. That's because on smooth muscle, CAMP will cause muscles to smooth muscle to relax. So beta 2 is excitatory because we're in the fight and flight response. We want our lungs to relax so they can be able to take deep breaths and get as much oxygen as we can so we can either fight the bear that's trying to kill us or we can run away from it. Alpha 1 is related to blood vessels, unless we're talking about blood vessels inside a skeletal muscle, then you're looking at beta 2. Alpha 1 or beta 1 is going to be the heart. Beta 3 is adipose tissue. Adipose tissue like fat. Not like it's fat. And beta 2, like I was talking about before, associated with lungs. If I remember correctly, M3 is going to be our gut. And then, yeah, so depending on if we're using an agonist, which will increase our signal, create an excitatory one. So let's say we use an agonist like atropine for a beta 1 that'll cause our heart rate to increase. So yeah, that's a good example. People use that in the ER to, if somebody's heart's flatline, then they'll give them atropine. I don't know if I'm pronouncing that right. And I believe that it kickstarts their heart so that it starts going again. We can also use an antagonist, which will create a different pathway. And then the relationship between these two, depending on which receptor we're talking about in this situation, combined with everything, we'll be able to improve or make different situations complicated. But yeah, that's what I've got.